So, thank you very much for joining us. Today we welcome you once more to Encontro Ciência. The talk will be recorded, so I ask anyone who does not want to appear to keep the video turned off. This video will later be available on CE3C YouTube channel. We are quite pleased to have today as our speaker Paolo Vaglini. Paolo will talk about his work. So thank you very much, Paolo, for being available to share with us your work. You can ask questions during the talk through the chat, or at the end you can ask Paolo directly. As I said, the conference will also be available on YouTube, so you can later also ask things to Paolo. Paolo will be presented by Teresa Diaz, our researcher. So thank you very much, Teresa. And now I'll give you the floor. Thank you.
Thank you, Patricia. Good morning, everyone. So it should be Cristina Cruz presenting Paolo, but unfortunately she cannot be here. So I will do my best to present Paolo. So Paolo, some of you know him as Pablo, but it's the same person. So Paolo is working with us since 2017, and he's doing a PhD in Applied and Environmental Microbiology here at the University of Lisbon. And he's been working within the Plant-Soil Ecology group. And he supervises Cristina Cruz and also Professor Rogério Tenreiro. And his PhD is very interesting. So he's working, the topic is very new. So he's working with plant growth promoting bacteria and how these bacteria can be involved in defense priming to induce systemic resistance in plants. His work is very interesting, and I'm sure that you will understand how interesting it is. But I would just like to call your attention to another aspect that he will not be showing you today. That is how he has been developing his soft skills along his PhD. Because when Paolo started, most of his experiments were just giving very strange results, or at the time they seemed very strange. And so Paolo was just repeating the experiments over and over again. But once we start thinking about the science in another perspective, it all made sense. And what Paolo is going to show you will make sense. Well, at least for us, it makes sense. So I hope you enjoy Paolo's talk. And Paolo, enjoy also this opportunity, okay?
Thank you very much for your kind introduction, Teresa. Mm-hmm. So I didn't need to say absolutely anything else, so I'll start straight away. I hope everybody can see the presentation. Okay, so in the next few minutes today, I'm going to talk about microorganisms and specifically all those microorganisms that are associated somehow to plants. And I'm also trying to share with you how can we exploit these microorganisms to improve the growth and the yield of our crops towards a more sustainable use of natural resources. So just to introduce you a bit, we most of us know that the food system and agricultural systems are facing unprecedented challenges nowadays, and this is mainly due to an ever-growing population, which is going to reach 9.2 billion people by the year 2050. This is almost 3 billion more than today. And this will entail also an increasing demand for food. I brought here the case of cereals, because cereals are strongly impacted by these environmental issues. And this is a matter of concern because cereals are the most cultivated crop worldwide. And they are considered as an essential source of nutrients for human diet, and so it's crucial in defining our diets, but not also our diets, also our cultures. However, today the production of cereals relies heavily on the use of chemical inputs in the soil, such as fertilizers or pesticides. And I just brought you here a chart in which we can see that in the middle of the 50s until the year 2000, the yield and the production of cereals increased. More than doubled in only 50 years, and this was a very good result. But we also see the negative effects of this improved yield, and as we know, is that in these years with the Green Revolution, a lot of chemical inputs with pesticides and fertilizer were used. So this entailed a great increase in production, but at the end, we realize today that these practices are maybe not the best ones in terms of soil health, and because we are degrading our land up to a point that almost 1/3 of the arable land has been lost just in the last 40 years. So these issues demand alternatives, and these alternatives have to be good enough to produce enough food for everybody, but they also have to maintain the quality of our agricultural soils. So in this context, microorganisms may come to our help. So we have to dig into the microscopic world to face these challenges, because microorganisms are really an integral part in the life of plants. And as I will tell you now, due to their great myriad of abilities, they can be exploited in order to reduce the chemical inputs in the soil and going towards more sustainable and microbe-based practices. So just to make you an idea, I included here a very simple scheme of the plant and its associated microbiota. So they are sharing such a tight relationship that we already know plants not as plants, but as holobionts, which is a unique ecological unit comprising the host plant and its microbiota. And in a very simple way, how does this work? Basically, plants confer a protective sanctuary for a myriad of microorganisms, and therefore they can multiply in a protected environment, which is also nutrient rich. And in exchange, to say in some way, they provide some essential function to the plant related to growth and defense. So with the starting with the unraveling of these with great populations of microorganisms, the plant microbiota has been studied and approached through different techniques within the plant science area. For example, from genomics, from the DNA studies, we now know much more about the microbial diversity that is inhabiting plants, but we also know about the functional genes that are within the plant and that belong to the microorganisms and that can be useful to exploit. Also from the area of metabolomics, we advanced a lot of our understanding regarding the microbial secretome. We know which kind of compounds they produce, which kind of compounds are shared also with the plant, and therefore how they intervene in the metabolic networks governing the communication between both partners. And lastly, but not less importantly, of course, we have microbiology, which has allowed us to isolate and screen and carry out many in vitro and in situ assays in such a way that we can test our microorganisms and screen them for their abilities to improve a given plant trait. So all these studies, which have been growing, especially in the last 30 years, have led to the formulation of what we call today microbial inoculants, which basically consist in a sole microorganism or a consortium of microorganisms that is used in order to be applied in the plant and in order to modulate the status of the plant towards increased growth or defense. So we are kind of, this can be similar to a vaccine, for example, in which we are vaccinating somehow the plant in order to cope better with future challenges. So this generally, this vaccination is broadly referred to as priming. This term is very broad and is used in many areas. But I'll try to explain what priming is in our context. So up to date, priming has been studied mostly from the point of view of the plant. And generally, we can define prime as the alteration of the status of the plant towards an increased growth or defensive factor. So as we can see here, priming has been studied mostly in vitro. So in vitro in the laboratory, we have seen many characteristics of inoculants, for example, their persistence, their capacity to form biofilms, or their production of a metabolic repertoire that can be useful for the plant. And on the other side, priming has been used or has been studied from the plant point of view, as I was saying before, with transcriptomics being probably the most familiar area in the field up to date. So we know that inoculation with microorganisms or priming agents can induce an alert state in the plant, which we refer as induced systemic resistance or systemic acquired resistance. And they can express a series of growth-related or defense-related factors that can somehow help the plants to respond in a more sustained manner to their surrounding environment. However, the effects of priming, especially the effects of priming with beneficial bacteria, have been grouped into a poorly defined concept of growth promotion, without really taking into account what is their mode of action, and especially how they interact with the myriad of microorganisms that are present in the plant. So we are convinced that sometimes the variable effects that we observe in microbial inoculants are due to the fact that they have not been approached from a holobiont perspective, but just in vitro or just from the plant side. So we are convinced that studying this interaction and studying how microbial inoculants can affect the microbiota of the plants can really help us going towards the formulation of microbial inoculants that take into account the joint action of not only inoculants, but also the microbiota and, of course, the plant. So during these years of PhD, I've been working with this kind of system, or this kind of procedure, and basically what I have the work is with seeds of wheat, and this is just a general material and method section, and I have germinated seeds, and these seeds, which I have here referred as NP, non-primed, have been divided in two groups, one of which remained non-primed, and the other one was primed with a plant growth promoting bacteria, which is our inoculant. So having established these two treatments, we isolated from these plants the bacterial community, and also we prepare some macerates of these plant tissues in order to then construct a series of combination as a proxy to study the effect of priming. So going a little bit more in detail, if we see the first part here, as a priming agent, we have used, I was saying, a PGPB, or a plant growth promoting bacterium, in this case from the genus Herbaspirillum, species Ceropedicae. And this is a well-known PGPB because, first of all, it was isolated from cereals, so it is good for this kind of assays. and is well known for its intimate association with plants, and they can promote the growth of plants in terms of nitrogen fixation, for example. And I also added another evidence here that we recently provided a couple of years ago, that we saw that the inoculation of this PGPB can reduce the oxidative stress in wheat leaves that are challenged with pathogens. As we can see here in the right leaf, it's less damaged because this leaf was primed previously with our PGPB prior to pathogen infection. So going through the methodology section, we also, in another work, we isolated the endophytes from non-primed and primed plants. And as we can see the short result here in the left side of the slide, we, as a surprise for us, we isolated a very different bacterial community from each treatment. So this motivated us to explore this community and to assess what is the potential of this microbiota and how they can interact with the plant growth promoting bacteria. In the last part of this method section, and that is we carried out a series of analyses that we are still using. And basically what we did was to employ an untargeted metabolomic approach in order to see the metabolic environment that is governing the environment of non-primed and primed roots. And not only, we also analyzed this series of combinations to explore what is the potential of the indigenous microbial community to metabolize or degrade or de novo synthesize novel metabolites in the endosphere environment, therefore in the root environment. So having this methodology in mind, this is a representation of what is our main hypothesis of the study. So following the scheme that we were showing before, from left to right, here is represented the holobiont, which is represented as a series of intricate metabolic networks, which are the result of a dynamic and constant communication between the root cells and the indigenous community that is inhabiting the plant. We hypothesized that when we added our inoculant, this will trigger a series of events in the holobiont, which will be mainly determined by a change or an alteration in the root signaling, but also, as we have just seen, to the alteration in the community structure, and also consequently to an alteration of the microbial signaling, and therefore this crosstalk between both holobionts will be altered somehow. And as a result, we will have a prime system in which the holobiont is primed, and we hypothesize that certain pathways may be favored. If we compare, for example, pathway Y here would be more potentiated with regards to the beginning, and others will be will decrease. Let's say will be less intense. So what I will now show you a series of maps explaining these complex interactions, and basically what we did was to show all the levels of the metabolic landscape in each situation, integrating several pathways that were released in the KEGG database. And basically we constructed 3 different maps, and I will try to explain briefly each one. So following the scheme of before, here we have, for example, the metabolism of linolenic acid and fatty acids. And the maps that we are seeing here are illustrating the metabolites that were founded within this pathway in non-prime plants and in prime plants. So we can, by eye, we can see that there are already some difference. For instance, this fatty acid arachidonate is, the levels of this acid are really increased here, but also this HPO3 precursor, that is an important precursor for the synthesis of several hormones related to jasmonic acid or cytokinins or azelaic acid. Also, this precursor is increased a lot. So this, for example, this different regulation, if we just focus on this precursor here, may be a hallmark of a prime state in which plants prefer, or primate plants prefer to accumulate precursors instead of directly producing a series of hormones. But this gets a little bit more complex now, because now here in this part of the picture, I depict what is the result of adding the bacteria from which they were isolated, and what is the repercussions that it has on the metabolite and the metabolic repertoire of the plant. So we see that, for example, the non-prime bacteria, the consortium of non-prime bacteria, de novo synthesize several precursors, like OPC8, for example, an important precursor for the synthesis of jasmonic acid, but also hydroxylinolenic acid, and also generally increased the levels of several hormones, like traumatic acid, which is a cytokinin, or hydroxyjasmonic acid, or 9-oxanoic acid, which is the precursor of azelaic acid. All these hormones, of course, are involved in important growth and defense-related processes. In prime plants, the result is slightly different. These bacteria, for instance, tend to consume this precursor much more than in the previous situation. And well, I will not, of course, talk about all the details for each metabolite, because this would get very long. And then I illustrate what the other two combinations of cultures here, what happens when the non-prime consortium grows in a primed environment, and vice versa. What happens with a prime consortium that grows in a non-primed environment. So this series of metabolic events, we as a proxy to study priming, we define them as the priming phase, because it's the moment in which the PGPR enters in action and triggers a series of metabolic rearrangements in the plant holobiont. So the same thing that we did with this map, we also constructed another map that basically combined the phenylpropanoid synthesis pathway with the terpenes, I think, or the flavonoid pathways. Okay. And of course, we also observed many differences which were dependent on the extract in which bacteria were grown, but also dependent on the consortium that was present in the system. This of particular impact is, for instance, that in the priming phase, the prime consortium increased a lot the levels of yangonin, and yangonin is, for instance, a cavalactone, and lactones are molecules that are important both in the precursor of heterocyclic compounds, but also in quorum sensing. Therefore, they have a big role in bacteria-bacteria communication. So this is indicating us that maybe the priming with the inoculant is somehow directing the bacteria towards different communication strategies. Also for gingerol, gingerol is a potent antioxidant, and its levels were very increased. But then, if we take a closer look, also other molecules were induced, like, for example, this glyciocarpine here. And glyciocarpine is an anthocyanin and is actively produced by the bacteria. And we know that anthocyanins no, not anthocyanins, phytolexin. It's a phytolexin, and is produced by a bacteria, but we know the function of phytolexins in plants. They are potent antimicrobial compounds. But here is the bacteria who is the responsible of this production of phytolexin, for instance. And this is very interesting because it was not the only phytolexin that was detected. And finally, we have this third map, which is combining the terpene metabolic pathway with the steroid metabolic pathway. I chose these maps because they are also involved in the synthesis of several hormones, such as we can see here, abscisic acid, brastinosteroids, or gibberellins. And as I was telling you before, the consortium, the PRIME consortium, again, increased a lot the levels of another molecule, which in this case is a furanone, and furanones are also involved in quantum sensing, especially for inhibiting bacterial signaling. And of course, I cannot describe everything, but we have also the new synthesis of other compounds that are related with not only hormones, but also phytolexins, toxins, or other compounds involved in plant bacteria communication. So this is finally, this is what our hypothetical model would be. That is very similar to our hypothesis. The image that we have is just only a few change, but this serves to confirm that actually the bacterial community that is inhabiting the plants really greatly determines the fate or the metabolic fate of the holobiont. So, for example, we have seen that in the unsaturated fatty acids generally tend to increase when plants are in contact with or are in relation with this community. And the same happens, the same rearrangements happen with hormones that are involved in quantum sensing, toxin, phytolexins, a series of metabolites that are that underlie many defensive and growth promotion processes. So this is a conclusion slide in which I want to share with you that we are going towards a deeper understanding of how microbial inoculants work, and with this, I hope, future publication, we will. Well, we actually submitted this, so now we have to wait. But we propose a novel mode of action for inoculants that do not comprise only an impact on the plant metabolism or transcriptome, but also an impact on the microbiota that is inhabiting plant tissues. And this impact entails not only a structural modulation, but also a functional modulation, in which the metabolic repertoire from both microbial and plant origin are shaped, are impacted, and therefore the holobiont status can be in another metabolic state. We don't know yet if this is a good metabolic state or a bad metabolic state, but what we hypothesize is that priming induced by these beneficial bacteria can really rely on these kind of modulations instead of only just being persistent in the system or other categorizations that they have suffered in the past. Regarding this, until recently, there was this idea that inoculants must persist in the plant system. They have to be very numerous and they have to be with the plant throughout all the plant development, but we don't see this thing. We rather see another mode of action of inoculants that relies on a metabolic imprinting in the holobiont, which is enough or necessary to drive plant metabolism towards different directions. So the role of inoculants as driving forces for plant growth promotion has to be redefined, or at least have to be explained, taking into account the plant microbiota. And I would like also to mention that these kind of techniques, therefore untargeted approaches in metabolomics, coupled to pathway search and mapping, can really give us a very complete picture of what is the situation, the metabolic situation of the plant in different moments. When a plant is primed, when a plant is challenged, when a plant is in normal situations. This has a real great potential, because if we apply this kind of methodologies in the pathosystem, we will have much deeper knowledge on how both the plant and the microbiota together respond to their enemies, and if these responses can be efficiently mediated by inoculants. So this is my last slide. This would be maybe the ultimate goal would be to go towards an agriculture that is microbiota assisted, in which the tripartite interaction and the crosstalk between the various players of this system will lead to healthy plants harboring healthy microbiota, and therefore more adapted or better adapted to respond to an ever-changing environment. And with this, I would like to thank you all for inviting me, of course, the first thing, and then my supervisors and the rest of the team that really participated in this experiment. I thank Professor Cristina Cruz and Rogério Tenreiro, of course, but also Professor Carlos Cordeiro and Marta Sousa Silva from the chemistry lab in the Faculdade de Ciências, as well as Anabela Bernardes, which contributed to the oxidative stress experiments, and also Giuseppe Piraccini, with which all the metabolomic studies started in Florence, here where I'm now, almost a year and a half ago. Thank you very much. I'll be more than glad to answer to your questions.
Thank you very much, Paolo. I'm just going to stop sharing for us to see.
Yeah.
Congratulations for your presentation. It was great. So we have lots of people clapping, and now I want to ask if anyone wants to ask some questions to Paolo, if there are some issues that you want to understand a bit better.
Can I ask a question, Patricia?
Of course.
Paolo, what do you think? So this priming effect is basically a change in the plant status, let's say. So if you were using a pathogen instead of a plant growth promoting bacteria, what do you think would have happened?
I think that I mean, it's known that plants get stronger when they get somehow vaccinated, even if they are good or bad. Because if they sense a pathogen and then they don't sense it, and then sense it again a few days later, they will respond also stronger. This is the difference between the acquired and induced resistance. Okay. The thing is that the advantage of the inoculants, I think, or bacteria that apparently do not damage the plant directly, is that they can increase or prime plants more gradually, without implying a very cost-effective use of defenses, for instance. Because if you use a pathogen instead of a neutral bacterium, let's say, maybe the onset of defensive responses is very big, but not so efficient. So I would expect that also pathogens work for these kind of things, but maybe neutral bacteria or bacteria that have been isolated from that tissue and everything, maybe can activate plant defenses maybe more efficiently. It's more a matter of maybe of efficiency. I don't know if I answered.
Yeah, but maybe it's more a balance between the changes in growth and defense.
Yeah.
If the plant is challenged with a pathogen, it will probably try to defend itself and not invest in growth, right?
Yeah.
So maybe that would be the big difference.
Yeah. Maybe the trade-off is small. Is a bit small. Yes.
Would you like to try it later?
Well, pathogen have to enter in the game in the next whatever it is, postdoc or I don't know, or the next victim that comes after me. I don't know.
Yeah, but I find Paolo's work very interesting, because this is almost like when we think about babies when they are born, if their birth is natural, so they get all this inoculation from the mother, and we really see a difference between babies that have a natural birth and a C-section, and this is almost a similar way of thinking of life in this case. So thank you very much, Paolo.
Yeah, thank you, Teresa.
So let's see if we have any other question arriving or not. I don't think we have, but of course the talk will be available in YouTube. So if you will see it again and if you have questions to ask Paolo, I think you are more than welcome to do it and to ask him through the email or whatever to see anything that you want to understand a bit better or to discuss with Paolo. So thank you very much for all of you for being present. I hope to see you in the next Encontro Ciência, and have a great week and weekend. Thank you very much.
Goodbye. Thank you, everybody.
Bye. Thank you, Paolo.